So today I want to tell you about some pretty exciting recent research. In 2010, Dr. Craig Venter, a scientist who has worked on the human genome and the diversity of life on Earth, made a big announcement. He and his team had made what they called the first synthetic cell in the lab. Okay, synthetic cell, what does that mean? Well, it doesn't mean that Dr. Venter and his group assembled a cell from scratch. That hasn't exactly been done yet, although there are many research groups that have made protocells, which have at least some of the parts of a naturally occurring cell. No, instead, what Dr. Venter's team had actually done was to construct a genome in the lab. A genome, if you remember, is the complete set of genetic instructions, and all cellular organisms have a genome that is composed of one or more molecules of DNA, deoxyribonucleic acid. So the team made a genome, and it was based on genomes that are already known and sequenced from naturally occurring organisms. It was mainly based on the genome of Mycoplasma mycoides, a bacterium that parasitizes cows, but they incorporated some other elements as well. They changed one gene, for example, in a way that makes the synthetic bacterium unable to stick to mammalian cells. Great idea, so that it cannot act as a parasite. They also put in sequences of DNA that act as watermarks, unique stretches of nucleotides that they constructed that could potentially be used to identify the synthetic genome in case it's ever found in another context. They also put in a gene called LAC-Z, which allows cells to make an enzyme called beta-galactosidase. And this enzyme allows cells to change a chemical called X-gal into a blue-colored product. And then they took a bacterial cell of a different species, Mycoplasma capricolum. They destroyed its genome and they replaced it with the artificial genome that they had made in the lab. And here is the amazingly cool part to me. The new genome completely worked. It took over and effectively changed the cell into a different species. And unlike the parent cells, the bacterial colonies were blue if they were grown on X-gal because they were expressing that LAC-Z gene that researchers incorporated into the genome. So it was a great reporter. These cells can grow and reproduce just like any normal cell. In fact, they exhibit all of the characteristics of cellular life. Okay, so the reason I'm telling you all about this fascinating work is because we need to talk now about what makes up cells. We've already learned that cells are made up of biomolecules and some of their important components, and now we're going to start diving deeper into some of those biomolecules. Remember when we discussed the characteristics of life and tried to come up with a way to define life? Well, as you know, we failed because there isn't one universal definition. Instead, we tend to talk about things that cells have in common and use those as traits that define life, or at least are hallmarks of life. Well, now we're going to dive deeper into that idea and look at cells. One way that some scientists think about cells, and indeed how to define life at all, is called the PMC model. All right, now that's not the only model, but it is pretty useful. So what does that stand for? P stands for program, M stands for metabolism, and C stands for container. Let's break that down a little more, and we'll start, of course, with the P. Okay, so the P stands for program, and this means the information that the cell stores and uses to do all of the things it needs to do, including metabolize and reproduce. This is what Craig Venter's group did by making a genome and placing it in a cell. In effect, they were rewriting the program of the cell with a program that they had created in the lab. So really here we're talking about DNA for cellular life. And if we wanted, we could also include the systems that we need to read and interpret the program. So this would include RNA, which has several roles in the cell, including making a copy of genes so that they can be expressed as proteins. Okay, next we have M for metabolism. Metabolism is all of the chemical reactions that take place in the cell, which is, pretty complicated, but we can sort of simplify it a little by thinking of it this way. Metabolism is about capturing energy and converting that energy into resources that the cell needs. Metabolism is carried out by many different types of proteins, and it would include doing things like taking carbohydrates, breaking them down in a controlled fashion in order to produce ATP, adenosine triphosphate, which is the energy molecule of cells. Finally, C, a container is a way to separate the cell from the rest of the universe. And we've already learned this because this is one of the 4 characteristics that all cells share, a cell membrane. All cells have a membrane, which is selectively permeable, meaning it lets in some materials into the cell and out of the cell, but not all. The membrane is made up of molecules called phospholipids, and in addition, it also has a lot of proteins embedded in it, which actively control what is going in and going out. Okay, so this is a useful model for us to think about and organize the materials that make up cells, as well as all of the things that cells need to do.